In Lecture 7, we're going to be looking at the process of protein synthesis. We know DNA encodes for the instruction to make the proteins, but exactly what are the steps involved, that's the focus of this lecture. So the first part of protein synthesis is a process called transcription. Before we learn about transcription, let's go through some terminologies. The genome of an organism is all of its genetic information. So if you sequence all your 46 chromosomes and figure out all the nucleotides in the chromosomes, that would be your genome. Here is a chromosome, and we have 46 of them. On each chromosome, there would be thousands of genes, and for simplicity, I show you 4 genes here. So the gene is a section of the chromosome that will have instruction to make a particular protein. Remember, proteins are amino acids that are connected together. So there are 20 amino acids. How do you know which amino acid to connect to which ones? That instruction is encoded within the gene. So gene A here might have the instruction to help you make insulin. Gene B here might have the instruction to help you make lactase, to break down lactose. And then gene C would be something else and something else. And you would have 2 copies of these genes for each chromosome, because you would have 1 chromosome inherited from each parent. So this is the paternal chromosome. This would be the maternal chromosome. So the location of a gene on the chromosome is called a locus, right? So think of the genome as like the entire textbook, okay, or like a cookbook or something. And then a gene would be a section in the cookbook that tells you how to make a chocolate cake. Okay, and the locus would be the page number that you can find the recipe on. I've been telling you that your chromosomes are inherited from each parent, 23 from each parent, right? So you will have 2 chromosome 1, you have 2 chromosome 2, all the way to 2 chromosome 22. And then for the sex chromosomes, you would either have XX as a female or XY as a male. So these chromosomes that are matched, so for example, this could be chromosome 2 from dad, and this could be chromosome 2 from mom. These chromosomes are actually called homologous chromosome, right? Because the homologous chromosomes are going to be same size, same length, and they are going to have genes in the same order. So if this is gene ABCD, on the other one is going to be in the exact same order. However, the version of the gene might be different. Okay, so let's say this gene here is going to determine your eye color, and this one you got from your dad, and is going to be blue eyes. Right? Whereas this one here, it's also going to be eye color, but you got it from your mother, and she happened to give you the brown eyes gene. Okay, so these two are both different versions of gene for the same trait, eye color. We call that allele. Okay, allele. Right. So along the entire homologous chromosomes, some of them will be identical. Some of the genes are going to be the same version. For example, if both of your parents have the same eye color, then this could be big B and big B. They don't have to be different, but there are going to be some variation because your mom and dad are not genetically identical, right? We talked about the central dogma of biology some time ago, and just to reiterate, what that means is we start with DNA, genetic material in the nucleus, and we are going to make a copy of that in the form of something called mRNA. Okay, so that process is called transcription. And after you create the RNA, we actually call it a pre-mRNA because it's not quite ready to exit the nucleus yet. So we're going to make some modifications to it in a process called post-transcriptional modification. Right? That's going to increase the stability of the RNA, and so after you do those modifications, it's going to be able to exit through a nuclear pore on the nuclear envelope, and then once it gets into the cytoplasm, you are going to be able to use that instruction to make proteins, and the organelle that is responsible for doing that would be the ribosome, and you would be doing it in a process called translation. So from DNA, it would go to RNA, and then it would go to protein. That would be the direction of the flow of information in a cell. I've been telling you that your chromosomes are inherited from each parent, 23 from each parent, right? So you will have 2 chromosome 1, you have 2 chromosome 2, all the way to 2 chromosome 22. And then for the sex chromosomes, you would either have XX as a female or XY as a male. So these chromosomes that are matched, so for example, this could be chromosome 2 from dad, and this could be chromosome 2 from mom. These chromosomes are actually called homologous chromosome, right? Because the homologous chromosomes are going to be same size, same length, and they're going to have genes in the same order. So if this is gene A, B, C, D, on the other one is going to be in the exact same order. However, the version of the gene might be different. Okay, so let's say this gene here is going to determine your eye color, and this one you got from your dad, and is going to be blue eyes. Right? Whereas this one here, it's also going to be eye color, but you got it from your mother, and she happened to give you the brown eyes gene. Okay, so these two are both different versions of gene for the same trait, eye color. We call that allele. Okay, allele. Right. So along the entire homologous chromosomes, some of them will be identical. Some of the genes are going to be the same version. For example, if both of your parents have the same eye color, then you know this could be big B and big B. They don't have to be different, but you know there are going to be some variation because your mom and dad are not genetically identical, right? We talked about the central dogma of biology some time ago, and just to reiterate, what that means is we start with DNA, genetic material in the nucleus, and we are going to make a copy of that in the form of something called mRNA. Okay, so that process is called transcription. And after you create the RNA, we actually call it a pre-mRNA because it's not quite ready to exit the nucleus yet. So we're going to make some modifications to it in a process called post-transcriptional modification. Right? That's going to increase the stability of the RNA, and so after you do those modifications, it's going to be able to exit through a nuclear pore on the nuclear envelope, and then once it gets into the cytoplasm, you are going to be able to use that instruction to make proteins, and the organelle that is responsible for doing that would be the ribosome, and you would be doing it in a process called translation. So from DNA, it would go to RNA, and then it would go to protein. That would be the direction of the flow of information in a cell. The purpose of transcription. DNA is too big to leave the nucleus, and it won't be able to fit through the nuclear pores, even if it wanted to leave the nucleus. Remember, during mitosis, when we try to move the chromosome around, the nuclear envelope actually needs to dissolve before the DNA can come out, right? So under normal circumstances, the DNA will not be able to leave the nucleus. And in fact, you don't want the DNA to leave the nucleus for no good reason, because they might get degraded in the cytoplasm. So how do you extract the information from the DNA to make the proteins? Well, to solve the problem, what we're going to do is we're going to make a copy of the gene that is needed to make the proteins in the form of mRNA. And that's what transcription is, right? So this adds a level of control. You can control when to copy the gene. You can control how often to copy the gene. And you're going to be able to turn on and off the gene by choosing when to copy it and when not to. For example, you're not going to have to make insulin 24/7, right? You only kind of need them whenever your blood sugar level is high. So there's no point turning on that gene all the time. And a lot of genes are going to be turned on a needed basis. And by having transcription, you can have that additional control. So recall there are base pairing rules in DNA. Adenine pairs with thymine, guanine pairs with cytosine. But in transcription, we are trying to make a DNA in the form of RNA. So the base pairing rules is similar for DNA and RNA, with one big exception. There is no thymine in RNA, and so A will now pair with uracil. So there are two strands of DNA, you should know by now, and the gene is actually going to be on just one of those strands. Okay, the strand that contains the instruction to make the protein, we call that the coding strand. The coding strand. Okay, that's where the instructions are. But we're gonna build our RNA using the template strand. The template strand. Okay, so the template strand gives you a mold, if you would, a template to build your RNA on. And once you've created the RNA is going to be a copy of the coding strand. So here is an example. Given the template strand, find the coding strand and the mRNA. So this one here is the template strand. So that's one strand of the DNA, and we know that the DNA is going to be double-stranded and complementary. So if I know what's on one side, I can easily predict the other side using DNA base pairing rule. So A goes with T, C goes with G, A, C, A, G. So it's just regular base pairing rule, Chagaff's rule, right? So that would be your double helix. Now we are going to do transcription. We are going to create a copy of mRNA. Okay, so what will the mRNA looks like? The mRNA is going to be identical to the coding strand, but complementary to your template strand. Okay, so we are going to have, instead of T, we're going to have U, because there's no thymine in RNA, right? So UUG, ACA, GCU, GAU. Okay, so the mRNA that you've created will contain the same information as the coding strand. Okay, because the coding strand is the one that contains the instruction to make the protein, right? But it will be complementary to the template strand. Okay, so that's why A will pair with U following DNA RNA pairing rules, and C will go with G, and G will go with C, so on and so forth. Let's try a different example. Create an mRNA from the given template strand. So this is template strand, and if I want to build an RNA from it, we will have to follow complementary base pairing rule. So A will pair with U for RNA, T will pair with A, C will pair with G, T will pair with A, and so on and so forth. Okay, so there is your RNA template, which again would contain the same information as the coding strand. Here's another example. Create an mRNA from the given coding strand. So this is already coding, which means your mRNA is going to look exactly like it, with the exception that T will be replaced by U. So I'm just going to switch T to U, and then I'm going to copy everything else. I'm going to copy everything else, but T will be replaced by U. Okay. Summary. The coding strand and the mRNA are basically identical to each other because the coding strand contains the instruction, and you want to make sure that the RNA is going to contain the exact same instruction for making the whatever protein you need. With one exception, that the uracil is going to be replacing all the thymine. So you can see here, I replace all the T with U because there is no thymine in RNA. The template strand and the mRNA strand are going to be complementary to each other. So where you have A, T, C, G, they are going to be paired with U, A, G, C, respectively. Right, so you can see if I give you the template strand, then you can switch it to the mRNA by following the complementary base pairing rules. So the actual process of transcription takes place in the nucleus of the cell, and the enzyme that's responsible for doing it is something called RNA polymerase. It's not surprising because we are building RNA, so we would need an RNA a polymerase, an enzyme that would help you polymerize RNA, basically. So the polymerase will pull apart the DNA strands, and then we will use the template strand to create the RNA strand. And the product of transcription is a strand of pre-mRNA. As mentioned earlier, the mRNA is not ready to go into the cytoplasm yet. It requires further processing before it's stable enough to go into the cytoplasm. There are 3 stages involved in the process of transcription. We have initiation, followed by elongation, and finally termination. In initiation, the RNA polymerase needs to find the beginning of a gene. Remember, the chromosome is really long and it has thousands of genes on it. How do you know where the beginning of the gene is going to be? Right? Think about a textbook. How do you know it is the beginning of a chapter? There are all words all over the place, right? But you would know the beginning of a chapter because there is usually a distinct page that says, you know, chapter 5 or something like that, and you would know that marks the beginning of a chapter. So in the same way, there's a special sequence called the promoter sequence that would mark the beginning of a gene. So here is the gene, and you know it's part of the DNA. And at the beginning, there is the promoter sequence. This promoter sequence is usually rich in adenine and thymine. Right? It's going to have a lot of A's and T's in it, and the reason for that is because if you look at the interaction between A and T, there are only 2 hydrogen bonds as opposed to 3 hydrogen bonds in C and G interaction. So only having 2 hydrogen bonds would make it easier for the RNA polymerase to pull apart the DNA strands. So the DNA polymerase will unwind the DNA strands once it binds to the promoter, and that would initiate the RNA synthesis process. In elongation, the RNA polymerase will be moving along the DNA strands. It will be unwinding the DNA ahead while rewinding the DNA that is behind it as it moves along the gene. And I want you to pay attention to this diagram over here. You can see the blue represent the DNA double helix, and you have the template strand here and the coding strand on top. So of course the coding strand and the template strands are going to be complementary to each other. You can see the T and the A pairs, and then another TA here. G goes with C, and so on and so forth. So what happens is the RNA polymerase will be adding new nucleotides on the 3' end of your mRNA. And it's going to be using the template strand as a template to create that new strand. So it will read the nucleotides on the template strand, and it will determine what the appropriate complementary nucleotide is going to be. So if it sees a T on the template strand, then RNA polymerase is going to put an A there. Okay? And then it sees an A there, it's going to put a uracil there, a U, and it will continue to grow. And what you end up having in the mRNA is that the nucleotides are going to be encoding the same information as the coding strand. Right? You can see CAT here, and you can see CAU here, right? Because we replace all the T with the U, right? And then CCA, CCA, T, and you have AAU here. So that's what we mean earlier when we say we use the template strand to create the mRNA, and it will result in an mRNA that is going to be containing the same information as the coding strand. So this is going to continue to happen, and eventually the RNA is going to reach the end of the gene, and the end of the gene is going to be marked by what's called the terminator sequence. Once you get to the terminator sequence, the RNA is going to rezip everything in the DNA. Right? Make sure the double helix is going to be wound up again, and it's going to fall off from DNA, and at the same time it will be releasing this pre-mRNA. In post-transcriptional modifications, we are going to add various modifications to the pre-mRNA to increase its stability in the cytoplasm. And in the process of copying the DNA, we actually copy what's called like some junk DNA. They actually don't encode for anything. So we want to remove those first before letting the messenger RNA out of the nucleus. So there are 3 steps that are involved in post-transcriptional modifications. We are going to be adding a 5' cap to the 5' end of your mRNA, and we are going to be adding what's called a poly A tail on the 3' end, and finally we're going to be doing something called the RNA splicing. So the 5' cap and the 3' poly A tail are going to increase the stability of the mRNA. The 5' cap is actually something that is made up of modified guanine nucleotides, and on the 3' end, you are going to be adding 50 to 250 adenine on it. That's why it's called a poly A tail. The adenine is the same as the nucleotide that you found in DNA, and both the cap and the tail will determine how stable the mRNA is. Okay, sometimes you don't need it to be too stable. Okay, you only need to make the proteins for a little bit, so you can create something that's a little bit less stable, and then it will break down after a very short period of time, because you don't need to keep on making that protein. In other times, you might want to make the proteins for a longer period of time. Then you would want to have perhaps a longer tail to increase the stability, and so it can hang around in the cytoplasm for longer periods of time. In RNA splicing, what happens is you will be removing introns, which are regions of DNA that doesn't encode for anything, and then you will be joining the rest of them together, and the rest of it are going to be called exons. So exons are the regions of genes that are going to be expressed. They are the regions of genes that actually contains the instruction to make the proteins. In the RNA that you've copied, right, there will be a bunch of introns in it. So you would take out all these things, and then you would connect the exon 1 with exon 2, and then exon 2 with exon 3, and then 3 with 4. And so you remove all the junk DNA, and you end up having a processed mRNA that is both stable and it doesn't contain any non-coding information. So this summarizes the whole process of transcription. Right? We have the RNA polymerase here going through our double helix, using the template strand here at the bottom to create the RNA transcript. That transcript will then be processed by removing the introns shown here in pink, light pink. You'll be adding the poly A tail and the cap, and now it's ready to exit through the nuclear pores into the cytoplasm. So in the next video, we will be looking at the process of translation, where the ribosome will be using the instruction on the RNA to create proteins.